On this week's Arabidopsis Research Roundup, I'm absolutely delighted to be joined by Mike Roberts from the University of Lancaster. And we're going to be talking about a paper just published in New Phytologist, the title of which is Jasmonic Acid-Dependent Regulation of Seed Dormancy Following Maternal Herbivory in Arabidopsis. So thank you very much, Mike, for joining us this morning. And it can be interesting to hear about this paper, which adds a little more complexity to what we know of an already very complex situation that controls seed dormancy.
So it's a whole new angle, as you say. So there's lots of information in the literature about abiotic regulation of seed dormancy, but this is one of the first real sort of detailed characterizations of an effective biotic stress.
Mm-hmm.
So the regulation of dormancy is important for plants to control, obviously, where the next generation start their life. So things like temperature and day length are particularly important cues in controlling dormancy, so that germination is timed to the right season. So there's lots and lots of, as I say, information on how that's regulated, and we know that the hormones ABA and GA, abscisic acid and gibberellic acid, are the key sort of antagonists. ABA promotes dormancy and inhibits germination, and gibberellic acid promotes germination. There are also various other hormones that interact with those processes and modulate things. And the idea behind this paper originally came from work that we were doing in my lab in collaboration with others on transgenerational immune priming, which is what we call an effect that we see where biotic stress, such as disease or herbivory in the parental generation, alters the defense phenotypes in the offspring generation. So the parents are passing on kind of defense priming to their offspring, so kind of warning them that there are potential pathogens or herbivores in the environment, and it enhances their defenses. So we're interested in whether there might be other kinds of maternal influences on the offspring depending on the environment. And what I've seen a few years ago from an old colleague in Graham in York was that they found that the compound OPDA, which is a precursor to jasmonic acid, is a regulator of seed dormancy. So there's a mutant that's been around for many years called comatose, which, as its name suggests, is very dormant, doesn't germinate very easily. And they show that increased dormancy phenotype is caused by increased levels of OPDA in the seeds. And we know when plants are attacked by herbivores that jasmonic acid is a key signal in those defense responses against herbivory. And we know that OPDA, the precursor to JA and various other jasmonates, are all elevated in tissues in plants under attack from herbivores. So I kind of thought it's one of these examples where I had an idea that might relate herbivory to dormancy, and we did some experiments, and it turned out that the general idea was right, but what I thought would happen was exactly the opposite in the end. So what I figured was that if OPDA increases dormancy, and plants under attack are making OPDA, then plants that are suffering herbivory would make seeds which are more dormant because of the elevated levels of OPDA. And that kind of made sense, because then you have this nice situation where, as well as priming the defense in the offspring, you also delay dormancy, so you wait longer before you germinate to avoid the herbivores that your parents were suffering from, and then when you do germinate, your defenses are primed. So we did the experiments to look at dormancy in plants that have been suffering from herbivory. What we found was that there was indeed an effect on dormancy, but it was completely the opposite effect of what we predicted. The seeds were actually much less dormant, so they germinated very rapidly. They were germinating basically as soon as they fall off the plant, but normally they need to have to ripen for a while before they'll germinate. To cut a long story short, we showed that this was related to the amount of jasmonates in the seed. We measured the seed, various seed hormones, in collaboration with Ian Graham's lab. So we know that after herbivory, there are increased levels of JA, importantly JA isoleucine, the main bioactive jasmonate, and there were increased levels of gibberellin as well, which was important, and reduced levels of ABA. So both the major hormones controlling seed dormancy were changing in the way that you would expect for seeds which were showing lower dormancy, which you obviously, and that the JA levels were up as well. And we did various experiments with JA mutants and showed that that dormancy effect was JA dependent. We couldn't see this herbivore or damage-induced effect on dormancy in JA mutants, so we know that the JA signaling pathway needs to be intact.
Okay.
We also showed that it wasn't just the levels of the other hormones that were important, but what we found was that the sensitivity to ABA was changed as well. So again, ABA normally is promoting dormancy and blocking germination. So firstly, we see reduced levels of ABA in the seeds, although the change wasn't dramatic. What was more dramatic was when we looked at the sensitivity to ABA. So again, if you break dormancy by its cold treatment stratification, which people who grow Arabidopsis are familiar with, we can make them all germinate even when they're dormant. We can break that dormancy by cold treatment. So we can do that and get 100% germination or close to 100% germination in our control and herbivore seeds from herbivore infested parents. If you germinate them on media containing ABA, you block the dormancy of the seed that came from the control parents. Okay. But the seed from the herbivore infested parents germinate just the same as if there was no ABA there. They don't see the ABA at all. Okay. And we did there's an even more dramatic effect with salt, so we put them on high salt, which again induces ABA signaling in the seed. And we can basically get almost zero germination in our control lines and pretty close to 100% germination in the herbivore stressed seed from the herbivore stressed parents. So there's a big change in ABA sensitivity, change in hormone levels. So we showed that there's this effect from herbivory via increases in the seed JA levels. And then Ian Graham's lab also happened to have some transgenic plants in their group, which they've not done a lot of work with yet. So these are plants where they'd produced a construct to drive expression of Jasmonic biosynthesis specifically in the seed. So they've taken a promoter from a seed-specific gene, fused it to alien oxide synthase, one of the key biosynthetic genes in the JA pathway, and put that back into Arabidopsis. So these plants make high levels of jasmonates in the seed, so similar to what we'd expect with the herbivore-infected parents that we've done the first experiments with. So we looked at those. They had similar changes in seed hormones and similar phenotypes, so they also had this reduced dormancy phenotype. So not only in the case of herbivory do we get elevated JA and a JA-dependent effect on dormancy, but we see that JA level, increase in JA level in the seed is sufficient to produce that phenotype. So the transgenic plants that make JA have the same reduction of dormancy and the same sensitivity to ABA. Okay. And interestingly also, because we were interested in defence, and that's where we sort of started thinking about these maternal effects when we looked at the defence phenotypes, and we saw this transgenerational increase in herbivore resistance in seed coming from parents who are infected with herbivores, which is what people have seen before. Right. But again, with the transgenic lines that are over-accumulating jasmonates in the seed, we also see a priming effect. So we now know that jasmonate accumulation in the seed is sufficient to generate this offspring priming effect.
Okay, so what we've known, I mean, classically we know that ABA, GA are the key hormones involved in germination control, dormancy control. But in this context, then obviously JA plays an important role as well. So, but in reality, in the real world, everything's also always under herbivory or pathogen attack. So in reality, JA would play a significant role in this process.
Potentially, yeah. So one of the interesting things is what the kind of biological role of this response might be, because as I say, initially we thought it might be a neat thing for the plants to increase their dormancy to escape herbivory, but the opposite's happening. So we don't really know whether this is beneficial for the plant or whether it's just something that because of the way that the JA regulation of ABA signalling's evolved in the past, that it's something they can't avoid. It's a kind of a negative consequence. There is some evidence. We've got some ideas that it might be beneficial, but we haven't tested them yet. So there's some evidence from other labs that under stressful conditions, precocious germination can provide an adaptive benefit. Okay. Because you're kind of out-competing seeds which are germinating later in the season.
Right.
So
I see.
Getting a head start in a stressful environment is better, basically. And again, if these seeds are also primed for defence, germinating quickly and getting going whilst you've got your prime defences could potentially give you an adaptive benefit. Interesting that what I don't know what I should say, this is one of those things where we also do some experiments that are not in the paper with disease, where you've got the opposite signalling pathway with SA, salicylic acid dependent defences, and there's very well known antagonism between JA and SA. And then what we see with seed from disease parents is that they're more dormant.
Okay.
So you do get the opposite effects from the JA. So that would kind of make a nicer story, but it doesn't be that way around in the first place, where you can say, well, in a pathogen-rich environment, you delay germination.
As long as the pathogens are of the same type each time.
That's the thing. You've got this trade-off depending on which defence pathway is more important. So again, it's kind of a similar situation with the transgenerational defence phenotypes, because there again, with the herbivores, you get transgenerational herbivore resistance, but you also get transgenerational susceptibility to pathogens and vice versa. Yeah. So it's all about the plants getting reliable information about the environment. So with the defence, we tend to see transgenerational priming only under very high levels of stress. If we just give moderate levels of disease or herbivory, you get priming within the plant, but it's not passed on to the next generation. You need much higher levels of stress. Right. So that's kind of, it's kind of like the plant measuring what the degree of stress is in the environment, and the higher the level of stress, the better the prediction is that stress might still be around the next generation, I guess. So it makes sense to prime the next generation.
Yeah, I see. So obviously this work is done in Arabidopsis and in different ecotypes of Arabidopsis, which have different levels of natural dormancy. But I know you do a lot of work with other plants as well, and JA, and using JA for germination studies. And we know that JA can have opposite effects in different plants. So do you think this effect will be consistent throughout other plant species? And are you looking in that in other plant species as well?
Yeah, so we've just got a few different species growing now that we're going to try the same kind of thing on. So we'll be talking to some seed companies, and there are particular crops that they have problems with dormancy in, and so they're quite interested whether this would be a potential way of kind of alleviating dormancy in some seed species.
Cool.
Seeds of some species which they have problems with. Right. So so we haven't got any information on that yet, but that's obviously a very natural next step to see how widespread the effect is.
And so the final question, in these experiments you use a spider mite for the herbivory experiments, is that right?
Yeah. That's right.
So do you see a similar effect, or maybe would you predict a similar effect if you just wounded the plants instead? So so if you just cut them with the scissors or something, or is there something specific that the mite is giving to the plant?
No, we actually do that in the paper. So I think buried in the supplementary information somewhere, an experiment on wounding. We did use that in the JA mutants, because the problem. So that was one of the most difficult experiments to do, because obviously the question is, when we saw this effect, is it JA dependent? And the obvious way you do that normally is with a mutant, but if you put the herbivores on the JA mutants, they basically die. Right. They get eaten to death because they lost all their defences. So we used wounding as a way around that. So we showed that would work in the wild type plants. We get a similar effect if we wound them during the flowering period. We can see this loss of dormancy. So we did that with the JA mutant. So that works. So.
Okay, I see.
So it's not specific to these kind of elicitors that specific signals of individual herbivores. It just seems to be more general.
Okay, great. All right, well, thank you very much, Mike, for providing a little discussion about this paper. And as ever, it shows that the interaction between hormones and environment, it's all about a trade-off between one thing and the other in order for the plant to maximise its growth.